In the beginning, I would like to thank Indian Academy of Sciences for giving me the opportunity to organize this symposium. And as he already pointed out, we have tried to give a various different perspective to the same system, namely molecular machines. So first 10 minutes, I'm going to give sort of an overview and go through the main thing of what molecular machines are, and then I'll point out which aspect of these are going to be picked up by the speakers in this particular symposium. So first thing is that, well, we look at around us and we find and we exclaim, well, it's moving, it's alive. So from well sperm to sperm well, movement of course is one of the hallmarks of life. And not only at this large scale, but even at the scale of E. coli bacteria or sperm cell, movement of course indicates life. And in fact, sometimes we believe, or we tend to believe, that plants probably are not moving, but plants do move. And people who investigated this include not only our J.C. Bose, but also Charles Darwin and so on. So movement is a hallmark of life. And so the question would be, then what sustains life? Well, what sustains life, the answer is just in one sort of sentence would be the molecular machines drive all the key processes which are indicators of life. In fact, at the root of all movements, the living system lies the movement at the subcellular and molecular levels. And this is just a cartoon of a cargo being transported by a molecular motor, which is somewhat like a porter carrying cargo. So cytoskeletal motors, forget about this term, cytoskeletal motors serve as porters, so they carry cargo over long distance, but motors do many more things. For example, there are motors which serve as sliders or rowers. So this is what happens inside muscle. So you have large number of motors which are driving sliding of the filaments, somewhat like the rowers who are actually moving this boat by working together. So motors can do all kinds of things. And so first question would be, what are the machines and motors? So for us, a machine is something which has an input, and this input could be matter, energy, or information, and it is an output. And if you have a specialized case where you have input in any form, but the output is mechanical, that means something is doing work, for example, against a load force, then we call these as motor. So now for all such machines, obviously as in our macroscopic world, fuel are needed, and it turns out that in these living systems, there are two major sources of energy. One is what is called NTP hydrolysis. Basically, there are some molecules called ATP or GTP, adenosine triphosphate or guanosine triphosphate, and they undergo a reaction which is called hydrolysis. So the details of this reaction is not going to be important for us. What is important that these reactions release some free energy, and part of that free energy is utilized for the movement of the motors. Of course, there is another source of this free energy, but that is not again going to be talked about in this particular symposium. Now, I have talked about sliders and rowers and so on, but there are other motors which are information processing machines, and this is related to what in molecular biology is called central dogma. So essentially what you have, you have a template. This template could be a DNA or an RNA, and you have machines which do three things. An example of such machines are what are called polymerases or ribosomes. What do? They do three things. They, like other motors, they move along a heteropolymer track, which is actually the template. They decode the genetic message, which is encoded in the template. And then third thing that they do, they polymerize a new heteropolymer using the heteropolymer on which they are moving as the template. So this, in fact, sometimes can be seen as even more interesting than just porters or rowers. In fact, there are all these machines that I have listed. You don't have to go through that list. So essentially, you have either what are called polymerases or ribosomes. They move along some template and give a product which is also a heteropolymer, and there are various different names associated with these processes. In fact, these kind of motors are going to be discussed in this symposium by Tanveer Hussain. Now, for polymerase motors, it is not the efficiency or power output which is so important. It is the accuracy which is more important rather than the speed. And this accuracy of template-directed polymerization is achieved at the cost of free energy dissipation. And in fact, this subject has also overlapped with computer science, because RNA polymerase are sometimes viewed as tape copying Turing machines. But again, this aspect is not going to be discussed in this particular symposium. Where are these motors or machines located? In fact, in this figure, you don't have to go through this. If you're interested, you can take a look. machines are distributed all over the cell, and of course they have different purpose. What is interesting that some of these machines actually don't just exist there. When they are needed, wherever they are needed, they are assembled, they do their operation, and at the end of it they even disassemble, and the parts of those machines are utilized for assembling other machines and other functions. So these are much more interesting than all the macroscopic machines that we are familiar with in the macroscopic world. So what is the basic principle? Basic principle is very simple. This is what the basic principle is. So there is a process which on its own will not go through. For example, if this is sitting on this particular plank, on its own it will not go up. However, when this comes down, this goes up. So they are always coupled processes. There is one process whereby the free energy that you gain, part of that at least, is utilized for driving something that will otherwise not go through. Say natural and unnatural one, these are coupled together, and that's how all these energy transduction processes of machines go through. Well, this is a mechano-mechanical, but because input output both are mechanical, then you can have chemochemical machines. Chemochemical, where you have a chemical reaction which goes from reactant to product. And this process, of course, releases free energy, enough of free energy that even part of this can be utilized for reactant to product going uphill in free energy. So again, two chemical reactions coupled, one of course goes uphill in free energy, and that is being driven by the driver, and the driver of course is bringing down the free energy. And what is the role of this object here? This is just the middleman. So this machine is essentially just taking energy from one, giving to the other, and this is the role of the chemochemical machine. So this was sort of popularized by Tedder Hill in this book several years ago. What we are interested in actually are chemomechanical machines. So these chemomechanical machines are taking free energy in the form of chemical energy input and doing mechanical work. So you have some molecule called ATP, which will go to diphosphate and inorganic phosphate. In this process, remember, it is different from the thermal machines that we sort of learn in our thermodynamics courses. Here it is not from high temperature to low temperature, it is from higher chemical potential to lower chemical potential. So this process is the natural process. This process then is releasing free energy, and part of that is being utilized for doing the work. Of course, this is at a finite temperature, it is coupled to a reservoir, and also it dissipates part of that free energy. So this is sort of the basic principle behind the operation of molecular machines. So one question. So these objects have nanometer sizes, tens of nanometers, and the forces that they generate are of the order of piconewtons. So natural question would be, is the mechanism of molecular motors identical to those of their macroscopic counterparts, except for the fact that you have a difference of scale. Instead of meters you have nanometers, instead of newtons you have piconewtons. If that were the case, it would have been boring. Certainly the emphatic answer is emphatic no. So there are many crucial differences between these motors or machines at the nanoscale with their macroscopic counterparts. So I have listed sort of what are the differences. So differences between man-made macro machines and nature's nanomachines. First thing is the molecular motors are made of soft matter, whereas our macro machines are made of hard materials to sustain wear and tear. Second, material components of molecular motor turn over at a high rate. Now of course, if that happened with a macroscopic, for example, cars, that would make it much more expensive. Then molecular motors operate in a sticky and noisy world. I'm going to explain in my talk this particular point. Molecular motions are driven by isothermal engines because they work inside our living cells at the body temperature. Driving and driven processes are loosely coupled. I'll explain it in my part of the talk. Namely, fuel consumption does not necessarily lead to movement under circumstances. Power stroke, which is so familiar in our macroscopic motors, are not necessary for molecular motors. They operate under conditions far from thermodynamic equilibrium. Unlike Carnot's engine, which I'm sure the students have seen in their textbook, okay? So unlike Carnot's engine, power output is non-zero, whereas power output of Carnot's engine is zero. Now there is a branch of thermodynamics called endo-reversible thermodynamics. This is not applicable in this case. Several distinct definitions of efficiency characterize different aspects. It's not just one definition of efficiency. When you talk about efficiency, you have to know which efficiency you are talking about. And accuracy can be orders of magnitude higher than thermodynamic bounds. most important here for the evolutionary biologists, that these are designed by nature through tinkering and Darwinian selection over billions of years, and not by, like engineers do, that they start from scratch and design something for a specific purpose. So now you must have seen ants carrying, each ant carrying, these are leaf-cutting ants carrying some leaf. Individually they're carrying this. But I'm sure you have also seen that apparently different ants, sort of as if they're competing, and still they succeed in carrying the cargo in a desired direction. Something similar happens at the nanoscale, and so a cargo can be carried by motors which are competing sort of against each other, and still the purpose is served. Now how this is happening, how this is done, is going to be discussed in Roop Mallik's lecture. Well, molecular motors, the name itself says that it's possible that sometimes there will be crowding and there will be traffic jams, and such things have been observed in the case of molecular motors too. Now, in just towards the end, I'd like to just tell you about the history of molecular machines. So to my knowledge, this term machines was first introduced by Marcello Malpighi in the 17th century, and he said that nature, in order to carry out the marvelous operations in animals and plants, has been pleased to construct their organized bodies with a very large number of machines, which are of necessity made up of extremely minute parts, so shaped and situated such as to form a marvelous organ, the composition of which are usually invisible to the naked eye without the aid of microscope. In fact, he was the father of microscopic anatomy. He started with the microscopy, but his microscopes are of course not adequate to see these machines, and we had to wait for 300 years to see those. And this concept of molecular machines was popularized by, in this particular article, by Bruce Alberts, who served at that time as the president of National Academy of Sciences US and later as editor-in-chief of Science. Essentially now we have two kinds of questions. Some of us are trying to reverse engineer natural nanomachines. These exist, we don't have the design. Nature has the design, but we don't have. So we have to find out what is the design. So that is one way of looking at it, as if like Charlie Chaplin, we are looking at this and trying to figure out how these work. And the opposite is the challenge number 2, which is forward engineering. That means we try to synthesize nanomachines. So this is that famous quotation from Feynman: what I cannot create, I do not understand. And these have been created, the beginning has been made, and that is what has already been mentioned. The Nobel Prize in Chemistry in 2016 was given for this purpose. Now let me just read out the delivering presentation speech on that particular occasion. Over the millennia, our society has enjoyed an ever-increasing plethora of useful machines for various purposes, in many ways leading to an enhanced quality of our lives. This progress has in particular accelerated since the Industrial Revolution. Today we are at the dawn of a new revolution that will bring us yet another giant leap forward. Okay, and what he's talking about is this new Industrial Revolution. I hope India will not be left behind, and academics will also take this up seriously. This is going to be the main topic of Pradyut Ghosh's lecture in this symposium. So these are the speakers in this symposium. And before I start my talk, I would like to thank Professor Ramaswamy, President of the Indian Academy of Sciences, for encouraging us to organize this, and the invited speakers of the symposium. So now I'll start my talk. So noise and non-equilibrium in nanomachines operation, physics perspective. So outline is as follows. As we do in physics, we look at mechanics, thermodynamics, kinetics, and then a few other things as I proceed. So first, molecular machine mechanics. Okay, I'm looking at motors only. So what is the mechanics? How is it different from the mechanics that we learn in macroscopic Newtonian mechanics? Well, I'm just quoting from this classic book in the 1942 edition, Dover edition, and even at that time, although nanomachines were not known, even at the micro scale, what he wrote down is really worth looking at. He wrote that gravitation in this domain, gravitation is forgotten. What he meant by that is inertia is negligibly small, and it is the viscosity of the liquid and the molecular shocks of the Brownian movement. Molecular viscosity gives rise to the viscous drag, and molecular shocks of the Brownian movement gives rise to random bombardments. These make up the physical environment. The predominant factors are no longer those of our scale. We have come to the age of a world of which we have no experience. Of course, that was 1942. Today we have a lot of experience of that, and where all our preconceptions must be recast. And what are the dominant forces on a motor? Well, dominant forces is external force. These are conservative force. You can define a potential. And this is, for example, you apply opposing force on the motor. So these are the external ones. And of course, it is working in a viscous medium. So there is a viscous drag force, which is dissipative force. And since it is working in an aqueous medium, the molecules of this aqueous medium, they are bombarding it from all sides. So that gives rise to thermal or random forces. As a consequence of that, if you look at the trajectory of the motors, so you will find that even if you start from the same initial point, every time you repeat it, you will find a different trajectory. So that is the consequence of the noise. So this can sometimes be called noise. So this noise force gives rise to these random trajectories. What about motor thermodynamics? The motor thermodynamics actually can bypass the constraints of the laws and can beat limits which thermodynamics imposes. How does it achieve? By operating far from equilibrium. So what are the fundamental questions here? So how do force generating molecular machines exploit noise? Now noise, normally we tend to believe noise is a nuisance. Can we sort of suppress, filter out noise? However, in this molecular world, it actually can be beneficial, and molecular machines can exploit noise for energy transduction. Now, does it violate the second law? We'll see that it doesn't. And then second question, that how do information processing machines achieve an accuracy which is far higher, orders of magnitude higher than the upper limits allowed by the laws of thermodynamics? So let me give an example, and this example has been chosen from our own work. So this is a motor. This is a motor. Unlike the normal porters, this is like a lame porter. It is one-legged, or actually called one-headed one. So this is called KIF 1A. It's called kinesin motor. It carries cargo. And what is observed, that it is like the distance over which it carries cargo is comparable to that of a normal two-headed kinesin, which are like normal porters. How does it achieve this? So this problem we studied, and what was done by our experimental collaborators is that genetically they varied the number of charged amino acids on this motor called KIF 1A and digested E-hook. E-hook is just one part of the track. Digested means they could cut it. So by basically changing the amount of charges on the motor and on the track, what was found that it is the electrostatic attraction between K loop of the motor and E-hook of the track that acts as a tether and enables one-dimensional diffusion of the motor along the track when it has taken fuel, namely ATP, it is hydrolyzing it, its affinity for the track goes down, so it detaches, but because of the electrostatic attraction, it cannot go very far. So remaining very close to the track, it executes diffusion, and at the end of this cycle, what is called ATPase cycle, wherever it finds it, it attaches there. So as a result, its step size is not exact 8 nanometers, what it should have been. It can be multiples of 8 nanometers, and not only it goes forward, it can also go backward, although it more often goes forward than backward. And this it is doing. How is it achieving this? Remember, it is during the diffusion it is moving. And what is the role of ATP? ATP basically allows it to detach from the track. So here it is exploiting thermal forces for its movement, and ATP is just actually helping it to go to that particular state. So this is indeed a physical realization of what is called Brownian ratchet. It exploits noise for its motility. Now obviously one would say, how is it possible? Well, it is possible because this system is not working in equilibrium, it is working far from equilibrium, where second law is not going to be applicable, and because of that it exploits thermal energy from the surroundings and executes directed movement. Okay, so I have discussed briefly the mechanics. I have discussed one example of thermodynamics, how it can beat thermodynamic laws by working away from equilibrium. Then let me look at some kinetics. And here the kinetics is not pure kind of movement only, because it is driven by chemistry. So it is coupled stochastic chemomechanical kinetics, chemistry drives mechanics, but there is noise coming from both mechanics and chemistry. Why? Because the force, I told you, force is random bombardment from all sides because of the thermal forces. So that is mechanical source of noise, but these motors work in environment where the concentration of fuel molecules or substrates is very low, and because of that there is also chemical noise. So it is actually combination of noise coming from both. So how do we actually describe them? So if you are not very comfortable with this equation, don't look at it. So basic idea is that one has to define states and rates. So you have to define what are states. So this is sort of the cartoon of that. So there are some discrete states, and what are the rates? System hops from one state to another, and these are the rates that have to be specified, and in terms of that we write the equations. And what is the thing that is evolving with time? What evolves with time is this probability of finding the particle. Particle in this state is just, here it is not a particle, it is just this object or system that is in the discrete state n at time t, and that changes as the system evolves with time. So this is written under Markovian approximation. Let me not get into the details of that. So now, how do we describe the, time evolution of the states? Well, the discrete mechanochemical states of the machine form the vertices of a network. You can call it a graph, while the directed edges of the graph denote the allowed transitions. So that's how we describe the evolution or the kinetics of the machine. So machine operation, that is its stochastic kinetics, is modeled as a Markov process in a heat bath at a given temperature, because it's the body temperature, and is formulated in terms of master equation. So this is for a typical machine. This is something that Tanvir Hussain is going to discuss in detail. So this is called ribosome. Now you don't have to know exactly what these details are. More important thing is that each of these discrete states have been labeled by integers 1, 2, 3, 4, 5. However, notice one thing, that it is not a simple cycle. There are pathways that connects the states directly going from here to here. That makes things complicated, and it has some consequences which I am going to discuss in a few minutes. Let me go back to Carnot's engine, which most of us have seen in school textbooks, and it should be described on the temperature entropy diagram, because then you have the thermodynamic states, and it has 4 different states. So it is isothermal expansion, adiabatic expansion, then isothermal compression, adiabatic compression. This is what it does. And in thermodynamics, when you say that it goes from 1 to 2, at what rate? Well, these are quasi-static. So it basically takes infinite time to complete one cycle. So finite amount of work done over infinite time, so that's why its power output is 0. Okay? Now, what we are considering is analogous here. So if you look at this cycle of course is a 5-state, not 4 states, but then there are some interesting things which differ from here. You have the rates, and here the rates were 0. Here you have some finite rates, and not only that, as I said earlier, that these machines have loose coupling. What does it mean? Well, for this particular machine, it starts from 1 and then goes to 2. While going from 2 to 3, it takes a molecule of GTP, which is a fuel molecule. It hydrolyzes it. So that energy is exploited. But then if it comes from this point to that point, this state to that state, it doesn't lead to the movement, forward movement. This is actually what is called kinetic proofreading, which is again something I will not get into in detail. But because of that, what happens? One molecule of fuel has been utilized but did not lead to any forward movement. However, it achieves something. This is the process in which it actually chooses its substrate very accurately. More often it does so, so more likelihood that it will choose the correct substrate. So then a price has to be paid, and the price is the higher the accuracy, reduce the speed. So it is this question, how did nature tune things in such a way that sufficient accuracy is achieved, at the same time the speed is also adequate for survival of the object. Now it's not only words, we also have to apply some theoretical techniques, and we apply network theory. Okay, now that theory that we apply was sort of popularized by Terrell Hill in this book, as well as Schnackenberg in this classic paper. So again, in words, what do we do? As I told you that we have these probabilities, we have to calculate those from these equations. How do we calculate them? We use a graph theoretic method, and from that we find out what are these probabilities in the steady state of the system. Now turns out that although it was popularized by these two gentlemen, the idea was actually given even earlier by chemists, and this is called King-Altman graph theoretical method in chemical kinetics. If you go back, again, King and Altman also rediscovered, and the original glory of derivation should go to no one than Kirchhoff. And I'm sure students also are familiar with Kirchhoff's laws for electrical circuits. And if you look at each of these nodes, where you actually calculate the voltages, these nodes are the counterpart of the probabilities of various different sites. And what about the currents? These currents are the counterpart of transitions from one state to another. So it's not surprising then that these concepts can be utilized for our calculations, and indeed I don't want to scare you with this. So basically applying graph theory, we can find out all the probabilities that I have shown in the previous diagram. But calculating just those probabilities is just first step of the calculation. What are the important things that we can calculate which are sort of can be compared with experiments? So first of all, remember that in such network of states, there are multiple pathways, each with multiple intermediate states with distinct transition rates. So what are the questions? One question is, what is the distribution of times taken for transition between a pair of states? This problem is actually in physics called first passage time problem. And how does this time depend on the topology of the landscape or the network? Another question, related question is, how does this landscape or the directed network get altered by physical force or chemical ligands, or it could be the reactions of the fuel burning, etc. And how does average rate depend on the force or the fuel concentration? The first passage time concept is very simple. So there is, say, a drunkard coming out of bar completely drunk, and then if you ask the question, how long does it take to reach its residence? Well, that of course is not a good question, because it can sort of overshoot, go further and then come back. So the relevant question would be, how much time does the drunkard take to reach the door of his house for the first time? And that is the concept of first passage time. For our random systems, of course, every time it does it, it will have different times. So you have a distribution of those times, and from that you can find the mean first passage time. And most often it is the inverse of mean first passage time that gives you the rate of the transitions. So let me give you some examples. Okay. So this is again for the same machine that I mentioned earlier, ribosome. And for ribosome, the dwell time distribution, which is basically the time it resides or dwells at a location before moving to the next one. So this was measured in these experiments, and this is the experimentally reported distribution of those dwell times, which are distribution of first passage times. And we calculated by our technique, and you can see that these lines, continuous lines, are obtained from the theory. The dots that are falling on these curves, they are the ones that we get from our simulations, and they are quite comparable to what was observed in the experiments. So the same cycles now again replotted here. So it is the same cycle, but now I am showing you how it moves from one codon, that is one side, to the next, which are labeled by the integer I minus 1, I plus 1. And what is dwell time distribution? Dwell time distribution is that it starts from here, you start your clock, and then how much time does it take to reach the next step? So this is the time that it has dwelt here. So that is nothing but the first passage time distribution, as I have said. And now I am asking the question that average velocity of this ribosome motor, which I was talking about, how does it depend on the substrate concentration? So in this case, the substrate has a name, it is called tRNA. We did this calculation, and we found that this is given by this particular equation, which chemists present here would identify that this is called the Michaelis-Menten equation. Okay, and Michaelis and Menten, okay, little more than 100 years ago, had considered this enzymatic reaction. Enzyme times substrate gives you a complex which can reversibly break, or irreversibly gives you the enzyme plus the product. And the equation for the rate of this, the average rate of course, is given by this equation, which you can see is very similar. So for us, the substrate dependence is being given by the Michaelis-Menten. However, if you look at what is called Michaelis constant and this Vmax, they are complicated combinations of those elementary rates of the various processes involved. Now of course, there can be errors in this process. So let me now look at the consequence of the errors. So this is the motor which is moving, and these are the substrate which come. Now there are two different stages where errors can occur. So there is something called charging on amino acylation of the tRNAs, where the amino acids are actually attached to the tRNAs, and there can be charging errors. On the other hand, the actual code that is there in the sequence of the track, which is called mRNA, messenger RNA, that has to be read by this object tRNA, which has anticodon, and on this you have triplets called codon, and codon-anticodon matching is crucial. Now of course, there can be error, and this is called reading error. So overall, there are various sources of error, and we sort of said, okay, how are they going to affect this rate that we have derived earlier. So this is what we had earlier. So this is one cycle as if there is no error, and now we are capturing all these errors in our model. So you will have 4 possibilities. You have a correctly charged correct tRNA. It is correct but mischarged. It is near cognate but correctly charged, and non-cognate but correctly charged. Now all this can be combined into one single network like this, and we calculated the average rate exactly. And this is even further generalized form of Michaelis-Menten equation. So the question that remains, when can we say that it will be Michaelis-Menten or generalized form of Michaelis-Menten? And this is exactly something that we are now calculating using graph theory, but I will not get into that in this talk. So now, reverse engineering of natural nanomachines. What is meant by reverse engineering? So in this case, so what I have talked about so far, that I had a theoretical model, and the forward problem is that from my calculation, then I find something which can then be compared with experimental data. But suppose you are provided experimental data, but you are not told what this machine is or what is the design, then going from the experimental data to the theoretical model is the inverse problem, and this is the problem of reverse engineering. So reverse engineering was defined very clearly by Rickoff quite a few years back. And reverse engineering is defined here as the act of creating a set of specifications for a piece of hardware by someone other than the original designers. Now of course, in our case, original designer is nature, and we don't have the design. So naturally, as far as natural nanomachines are concerned, this is what we have to do. We have to reverse engineer. We have to try to understand what is the design, not only the structure of it, but also the software, how does it work. Okay, so that is what sort of we have been doing. So first question here would be that how can the stochastic kinetic model of operation of a nanomachine, that is the number of nodes, remember this was like a graph. So how can we find the number of nodes and the rates of transitions indicated by the directed links of this network be extracted by methods of reverse engineering? Let me take one very simple example. It's a hypothetical one. You have a cycle. Just imagine that you have some machine for which, suppose this is the cycle. So what are the simplifications? Well, there is no branches coming out of this. So this unbranched cycle. This is fully irreversible. There is no backward, going backward. All is going forward. And the rates are all identical, k, and suppose there are n such nodes. Now, of course, we don't know this. What we have from experiment are the dwell times. How much time it spends at a location, how much at the next. And this is of course random, so you have a distribution of those, and from that you find out the t square average minus t average square, which is the fluctuation, and you normalize by mean square. So let's call this randomness parameter. This concept was introduced by Schmitzer and Bloch quite a few years ago in the context of molecular machines. It turns out that in atomic and molecular physics it existed many years ago called Fano factor. Okay, so now for this particular cycle, you can find out exactly what is the distribution of the dwell times or cycle completion times, and this is given by this distribution, which physicists normally call gamma distribution. But again, as it happens so often, that this was many years ago, in fact more than 100 years ago, this was discovered for the first time by Erlang in the context of queuing theory. That's called Erlang distribution. One feature of this distribution is that if you now calculate this randomness parameter, you find that it is 1 by n. What is n? N is the number of nodes in this. So now if an experimentalist has the distribution of the dwell times, from that one can get the number of nodes, although that is not available to us a priori. Unfortunately, what happens that nature is not so simple, and because of that it only gives you a bound, it doesn't give you the exact number. Let me skip this. Okay. If you have some nodes which are hidden, you don't see it because of fast measurements, what to do? Well, this is one example. This is for the same motor ribosome that I was talking about. I'll not get again into the details of what they represent. Experts can see it from these figures. And for this, the cryo-electron microscopy done by our collaborators, Joachim Frank, okay, they showed that there are 4 states involved, whereas another collaborator, Ruben Gonzalez, who does single molecule freight, he says that there are 2 states only, these 2 states. So he doesn't see 4 states. How do I reconcile the 2? That means it's possible that the 2 states are hidden because these are short-lived. So question is, can I have a better measurement that I can detect them? So naturally some quantitative analysis is needed, and that is something that we did. So we had a combination of equilibrium statistical mechanical calculation that we did, and we also applied mean first passage time technique. I'll again not get into that all the details. So what we showed that there are indeed two short-lived states which were picked up by cryo-electron microscopy but could not be picked up by single molecule FRET. And the reason is the CCD camera that you need to pick up these states has to be 10 times faster than what is available, but hopefully someday they are going to be picked up. Now you may say, why should one worry about those? Well, later if anybody is interested, I can explain why one has to look at those states, otherwise some physics may be lost. And then traffic-like collective movements, I told you that motors move on a track and can be crowded and can give rise to jams. So we have sort of applied this technique of what is called totally asymmetric simple exclusion process for such understanding such collective phenomena. So you have something called polyribosome, many ribosomes, those motors moving simultaneously on the track, and we have developed a formalism for understanding the formation of these polysomes. So I'll not discuss again. Same kind of concept can be applied to another phenomenon called transcriptional interference. Okay, so that again large number of motors, in this case these are different motors called RNA polymerase, which are moving on a DNA track. And very recently we have applied the same kind of formalism to intraflagellar transport, where you have what is called IFT trains, intraflagellar transport trains, which are pulled by motors, and we have applied again the concept of totally asymmetric simple exclusion process, which yesterday was introduced, also sort of explained in his talk by Arvind Ayar. So there are phenomena called programmed error, again done by these machines. So these are not random errors. Nature has introduced this deliberately, and one such phenomena is called internal ribosome entry, and that again we have studied using the same technique that I mentioned earlier. And frame shift is yet another programmed error, and for that we have developed a theory. Again, I don't have time to go into all that. Before I end, I'd like to acknowledge I have had collaboration with many people from various different institutions all over the world, and I had a large number of students. Although I had never done biology in school, college, university, anywhere till the age 45 when I decided to move over biology. So only in the last 15 years, this is what I have done. And I have been generously supported by various agencies. Thank you for your attention.
Questions? Arvind.
So, just one clarification. In your 5-chain process, are your rates fixed quantities that depend on internal parameters of the cell, or are they stochastic processes?
Okay, those rates are taken from experiments, but one of the rates, the first one, is proportional to the tRNA concentration. Okay, and there are two others which in principle are governed by the GTP concentrations. There are two molecules of GTP which are hydrolyzed, the first one for kinetic proofreading, second one for translocation.
But are they fixed numbers?
Well, as far as the analytical expressions are concerned, these are alpha, beta, gamma like thing, right? And then when you apply it, so these are the numbers that we take from experiments.
When you look at motor action on nanoscale, are quantum effects ever important?
No. If you look at the frequency that will be involved, that is too high. So if you look at KBT and with that, what will be the H nu? So if you equate that, the nu that you will get, the frequency is too high, and that will be washed out at this body temperature. So at body temperature, that is not important. So whenever you have various conformations where weak interactions are involved, it is not important. But however, if you look at the backbone, that of course is all quantum. So if you look at the proteins that make these machines, the making of the protein through covalent bond, that is quantum physics or quantum chemistry. But as far as the other things that I have talked about is concerned, it is dominated by thermal fluctuation, not by quantum fluctuation.
Okay, Debashish, where does the volume of the confined cell, all this is happening inside a cell, inside the nucleus and so on. no, none of your expressions seem to involve the actual physical volume. So, the effects of noise are exacerbated by the small volumes and...
Yeah, so most of the comparisons that we make are with in vitro experiments. In vitro experiment outside the cell. If you put it inside the cell, not only that, you will have to put volume, but more importantly, there is a phenomenon called crowding. So concentration of each species is very low, but there are a large number of other ones, so those have not been put in. So most of these comparisons are with experiment in vitro so far.
Sir, you mentioned about using the Michaelis-Menten equation in the ribosomal context. Sir, my question is, how do you see those assumptions, original assumptions in this, in this application?
No, we don't use it. What we do is that we have the model guided by experiments. From that we calculate the distribution of the dwell times, which of course depends on the substrate concentration, which is tRNA. Then we find out what is the mean time, and the inverse of the mean gives me the rate of this reaction. So this is very similar to what is done in single molecule enzymology. So there you have just one single enzyme and you don't have a rate. What you have is the turnover times. So one substrate comes out, after that there's a gap, another substrate comes out, and usually these are fluorescent. So these are fluorogenic ones. So you have the time gap between the successive fluorescence pulses, and then what you ask, that of course what Michaelis-Menten did for rates is not applicable directly, because you are having just discrete times after which products are coming out. But then from this distribution you can calculate the mean, and then that mean, inverse of the mean, will give you the rate. You ask the question, does the mean still follow Michaelis-Menten? And for many enzymes it is still valid. So it is in that spirit that we are doing here, that we have discrete ones, events, and then we have the distribution. We find out the mean, and the inverse of that is giving us the rate, which happens to be Michaelis-Menten. Now why it is so, under wide variety of circumstances, is something we don't know. We have actually almost completed a graph theoretical analysis proving a theorem in collaboration with my collaborators from Harvard Medical School, and so that theorem combined with our ones actually is going to settle this question. We are writing it up, that's why I cannot tell you now. So that will answer under what circumstances it will be Michaelis-Menten or generalized one, and under what circumstance it will not be so. It depends on the connected net of the graphs of a particular kind.
Actually, a very short clarification. You said there are two things. One is converting the Brownian motion or the noise into translation, and the second is to have the non-equilibrium into a finite power output. Are these two connected? I missed the link.
No, so second part I couldn't understand. First part, yes, that noise, that it is just like you are moving a sheep.
Right.
Okay, and you are not giving any energy to it, as if the sheep is taking energy from the surroundings and moving. Okay, but that of course would violate the second law. The point here that in every cycle you are pumping in energy, dissipating it. So it is, you are making it, you are always keeping it away from thermodynamic equilibrium.
Right. So these two are connected phenomenon.
Yeah.
Oh.
Yeah, it's a very good presentation. In fact, it was reminding me of whether messenger RNA moves or ribosomes moves. But having said that, when we understand the molecular machines, is there anyone also trying to move back to biomedical importance? Like using this, one can find out
of course, see,
inhibiting the movement and trying to have, say, spindle fibers.
One thing that my collaborators routinely do, that instead of GTP, they give GTP analog, which is non-hydrolyzable. So, then in the translocation, it just gets stuck. Okay, so that is done routinely. Now, for the purpose that you are asking, I am not the right person, I'm a physicist, but I know that there are many people, including past Nobel laureate Ada Yonath, who actually does a lot of work on this kind of things.
Okay, please join me in thanking Professor Chowdhury for a very interesting talk.